first part of the essay, we are going to define homeostasis. All right. Homeostasis is the body's ability to maintain an internal environment despite changes in the external environment. These are things such as body temp, blood glucose, and things like how much water is in your body, so I'll put water retention. Water retention. Okay, so with these, the way homeostasis is regulated in the bodies is we have a set point, or a point that the body is trying to maintain. For body temperature, the set point is roughly 98.6. Blood glucose is about 90. We want that at about 90. And so on and so forth. So the way this is done is we have certain receptors. The receptors send a signal that they sense from the stimulus. So the stimulus comes in, the receptors will sense it. They'll send a communication through afferent pathways. So we'll put afferent pathways to the control center. Then the control center, it will send a signal through the efferent pathways to the effectors. And those effectors, they're the things that have the ability to change the variable that we are measuring. So, an example of this. Let's go through an example of homeostasis and how your body regulates it. Okay, so let's talk about water and how it's retained in the body or sent through, and you pee it out. Okay, so you have receptors that will tell your body, hey, we need more water. So the receptors say more water. Okay, and then the receptor will activate an afferent pathway. And this, it's a pathway up to the brain to say, hey, we need to retain more water. So then in the control center, it says we need to retain water, and it will process that. And so need to retain water. And it will send a signal down the efferent pathways in the form of ADH, the antidiuretic hormone. So then the effectors, which in this case would be kidneys, would retain that water. So then this would retain retain water and bring the hydration or the water content of the body back up towards the set point, the point that we want it to be at. That's a quick example that shows how it moves. There are different pathways that we use and different feedback loops. So the first one that we use is a negative feedback loop. So what this is negative feedback loop takes the variable that was measured and it makes it go back towards the set point. It opposes any change in the internal environment, happens to the internal environment due to the external environment. So a good graph to show this, we have some sort of variable here, and this is time. So our variable will switch colors to red here, has a very certain set point. So this will be our set point, the point the body's trying to maintain. And then it'll have a range. And this is the normal range. There's the normal range. Okay. So your average negative feedback, if you were to represent it on a graph, would look something like that, where as the variable is getting farther from the set point over time, there are effectors in place to bring it back towards that set point. And again, it gets farther away from the set point over time. Effectors bring it back up. It doesn't matter if it's going up or down, as long as the variable is moving back towards the set point and opposing the initial change in the internal environment, we call that a negative feedback loop. The other feedback loop that we have is the positive feedback loop. So the positive feedback loop is a feedback loop where it amplifies the initial change in the variable. So the graph of that could look something like this. We have our variable over here on that axis, and then over here we have, again, time, variable and time. So we have, again, our set point here, the normal range here and here, and then in most cases we have a lethal range. If we get outside that range, it could be potentially lethal. So in this, we have our variable, and we can say that it's acting normally at first. The effectors are working, so it's doing its thing here, and then it can go into a negative feedback loop that is not working properly, and so it can continue trying to regulate, but it keeps climbing, and once it hits a certain point and everything else stops, it's going to amplify and continue up. That is where the positive feedback loop happens. It just keeps adding to that initial change. And there's very few of these that are actually beneficial, one of which being oxytocin release during childbirth. That is a beneficial positive feedback loop. The next part of the essay is different types of protein-mediated transport. And to understand protein-mediated transport, we first need to talk about diffusion. So diffusion is the movement of particles across the membrane from areas of high concentration to low concentration. So we have a high concentration to a low concentration. If we have small molecules that are relatively uncharged, nonpolar, they can go through simple diffusion. So simple diffusion, put it up here, is for small uncharged particles. And they will often, we have the membrane here, the cellular membrane here, they will just diffuse from an area of high concentration here to low concentration. They'll move down their concentration gradient. This doesn't require any energy or really any channels, it just goes. The next part is for facilitated diffusion. Facilitated diffusion. This is the type of diffusion that still requires no energy is required. That still goes from high concentration to low concentration. However, where this one differs is for this we need some sort of transport protein, some sort of membrane, intermembrane channel. Okay, so this must be facilitated by membrane protein. Okay. All right, so this is for anything that is a little bit bigger, could carry a charge, could have anything like that. They need some sort of protein channel to go through. A good one is aquaporins. So if we have a cell here and we have a bunch of water particles out here that need to go into the cell, we'll need some sort of way to get through, because as the phospholipid bilayer exists, the outside polar heads are hydrophilic and the inside is hydrophobic. So water will not pass through the membrane without help. So in order to do this, we need these aquaporins for water polar molecules to make it across the membrane into the cell. This is a form of facilitated diffusion. It does not require any energy. We're going from concentration, high concentration to low concentration. But in this case, we need some sort of facilitator protein to help us get the particles through the membrane. Now that we understand those, the next part are the other types of facilitated diffusion. So these will be our channels. So we have channel proteins, and these channels, like the aquaporins, are things that allow things in. we have three different types of channels that are located inside the cell membrane that allow things to come in. We have voltage gated channels. We have mechanically gated. We have ligand gated. Okay. So with these voltage gated channels, these are channels that have some sort of gate or mechanism that keeps them closed under most conditions. These will open when there is a change in the voltage or membrane potential or some change in the voltage. So that's when they will open, and they close by similar things. Change in voltage will close them. Mechanically gated, these are a lot of our touch receptors and things like that. They'll change with movement or mechanical changes in the cell membrane itself. So we will say changes in membrane. And these ligand gated, this last type that we're looking at, these will have some sort of substance that will come and bind to the channel to open it. So we have a ligand that comes and binds to open. So these are still just diffusion. They're facilitated diffusion. They're ways to get things into the cell, but they have some sort of gate that needs to be opened. And the way that those are opened is either through voltage, mechanically, or some sort of ligand coming and hooking in. So, yeah. There are certain things that will affect the rate of diffusion. I've mentioned a couple of them, but the rate of certain membrane proteins and their diffusion across they have only so much that can go through. So the rate can look something like this. So this is the concentration, and this is the rate. As concentration goes up, rate goes up, but only to a certain point, and then it'll level out. This is because the facilitator, whatever's moving things across the membrane, will become saturated, which means it can't accept any more coming in. It's hit its max that it can accept and funnel across because it doesn't have any more space to do so. This is our saturation, how saturation affects it. Another thing that can affect it is competition. There's certain transporters in the body that have similar molecules that will bind to them and compete for the transport across membranes, or drugs that will come in and bind to the gates or the different carriers and won't allow for things to get through. And that can slow the rate as well as far as competition goes. And most of these, in fact, almost all of these different transmembrane proteins, these channels, are specific. We can have our potassium channels, our sodium channels, our aquaporins. They're specific to the type of molecule that gets through them, that they will bring into the cell. And the direction is dependent upon concentration. It always goes from high concentration to low concentration through these diffusion. Diffusion is always from high concentration to low concentration. Now we will get into active transport. Active transport it has two different types. It has primary active transport and secondary active transport. So, primary active transport. Primary active transport it directly uses ATP. Or GTP, but in this case we'll say ATP. And it moves things against the concentration gradient. So it will go from low concentration to high concentration. This is why we need the ATP, the energy, to force things up their concentration gradient despite them wanting to move down their concentration gradient. A great example of this is the sodium potassium pump. Sodium is concentrated outside of the cell, but in the sodium potassium pump we're taking 3 sodiums from the inner cell and putting them outside the cell, and 2 potassiums from outside the cell and taking them inside the cell, which is also against its concentration gradient. And that pump will directly use ATP to accomplish this. Secondary active transport, let's do secondary up here. Secondary active transport, it also goes from low concentration to high concentration. However, it does not use ATP directly. So we're going to say no ATP. However, it does utilize concentration gradients that are established by ATP, such as the sodium gradient. We'll use that as an example. So the sodium gradient can be used to bring things in or out of the cell. We use these words symport and antiport. So symport is when, let's say, we have a cell here and we have some sodium out here, some Na, and we have a little sodium in here. So it wants to go back down its concentration gradient. So if we have another molecule out in the extracellular fluid that needs to come in, we'll just draw a dot for this one. One dot out here, and we'll draw a few in here as an example. If we want it to come in, symport is there going the same way. As this sodium comes in with its concentration gradient, this other molecule will cross with it. However, it will be moving against its concentration gradient, making this a form of active transport. The other type that we have is called antiport. Antiport would be the exact opposite. So we have low sodium in the cell again and high concentrations of sodium outside the cell. And inside the cell, we have one of these other molecules, and outside they're numerous. They're everywhere out here. In antiport, as the sodium goes down its concentration gradient into the cell, this other molecule, whatever the other transport is, will come out of the cell into the extracellular fluid against the direction of the sodium. And that is antiport. And those are the two forms of secondary active transport. And that should cover everything for protein-mediated transport. The last thing that we're going to cover is osmosis. Osmosis is the movement of water across the cell membrane. So osmosis, the water usually moves from places with lower concentrations to higher concentrations. And we measure this through osmotic pressure. That can be measured in osmoles. And osmoles is particles per liter. This is different than molarity, because molarity is compounds or molecules, that sort of thing. Here we have particles. So we could have salt, sodium chloride, NaCl, in 1 molar. 1 molar of NaCl in a perfect world, not our world, but in a perfect world, would become 2 osmolar, because it would split into 1 osmolar of sodium and 1 osmolar of chloride. That is osmolarity. So, with osmolarity, that is what makes osmosis move. So if we have a cell here with an osmotic pressure of 300 milliosmoles and the extracellular space has 250 milliosmoles, the osmotic pressure is pulling water into the cell. And this is to try and maintain a state of equilibrium. So what will happen is water will go in through the aquaporin in here. And this is usually driven by particles. As said before, the higher the milliosmolarity, the higher the osmotic pressure, because there's more particles there. An idea with molarity is the idea of tonicity. So we are going to make a little list, a little diagram here. We have osmolarity and tonicity. So if we were to draw a cell here and have it be 300 milliosmoles on the inside, and there was some solution here that was 300 milliosmoles, these would be isoosmotic, because the osmotic pressure in both are the same. However, if we were to have a cell with 300, we'll keep that standard, and some solution with 250 milliosmoles, this solution would be hypoosmotic, hypo being lower. So in this we would be hypoosmotic, and it would be the same the other way. If we had a cell with 300 milliosmoles and some solution out here with 350, would be hyperosmotic. Hyperosmotic. osmotic. Now this differs from tonicity because where this is a comparison of the osmotic pressure, we have the same osmotic pressure and then differing osmotic pressures. Tonicity is the effect that it would have on the cells. So if we were to take these two cells and put them together here, so we put the cell in the solution and it's 300 and 300. These would be isoosmotic, as it said before. They would also be isotonic because there would be no net flow of water into the cell, not making it swell or shrink. Tonicity is the effect that the pressure and the flow of water has on the stretch or shrinking of the cell, the outer pressures of the cell. So this one here, put that there, we have 300 and 250. So these are hypoosmotic, and if the water would flow into the cell to try and maintain that gradient and make it hypotonic. So we'll go hypotonic. And this bottom one, we have this. The net flow of water would be out of the cell, making it shrivel if we just kept it 300 and 350, and that would be hypertonic. Now, in these examples, it just happens that they match up and the water would do what it says. So the hypo one, it would flow in and swell up the cell. However, that isn't always the case. There's certain molecules and things that will change the tonicity and the osmolarity. So if we have this solution here with a cell inside of it, and the cell, we're just going to keep 300, right? But we're going to represent it in other ways as well. We're going to say 300 milliosmol. But we can represent those by these 4 red dots. Here, 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 and here. Well, the outside solution is 300 milliosmoles of the same kinds of stuff, kinds of things. It's going to be a list. So they would be isotonic because they're equal. There's not going to be any net flow. There's not going to be any flow of water, net flow of water in or out of the cell. It's all going to be at equilibrium. So we would call this isotonic. However, if we were to use something like glucose out here, glucose is taken down its concentration gradient into the cell and metabolized almost instantly. So at first, it would be in isoosmotic solution. Everything is the same. But eventually, these would go in and be metabolized, making this out here, this osmolarity, 0. Because it has no particles out there. So that would cause water to flow into the cell, making it expand and swell and be a hypotonic cell, and it could burst. But this is where it can get interesting. If we have both the .9% saline and the glucose in here, just draw them both right there like that, this would be a hyperosmotic solution because there's more osmotic pressure outside than there is inside. So in this, we would have a hyperosmotic And this outside, the extracellular, would be 600 milliosmol at first. But the glucose, like I mentioned, would go into the cell and be metabolized almost instantly. This would cut that from 600 down to 300 right away and make this solution isotonic. Because the glucose would move so fast that the cell wouldn't really recognize it, and it would equalize at 300.